Okay, so there's an enormous amount of material to get through. The most important thing that I wanted to talk about today is the sensory and motor homunculi of Wilder-Penfield, which is at the end of the lecture here. We'll see if I can make it to that. Anything above that's gravy. And I'm going to kind of gloss over some stuff here that normally I give a little more air to. I've cut like 3 lectures out of this neuronal sequence that I normally give, I guess. So today we're going to begin at least by talking about structural and functional classifications of various neurons in the CNS and PNS. Last time we talked about functional categories of afferent neurons. Now we're moving from the afferent into the central nervous system. So here is just a morphological or structural classification of neurons. Well, first, let's just go through them, I guess. Anaxonic neurons is the first category. These are axons, or these are neurons that do have a single axon. However, it's hard to tell them apart from the dendrites. They look like kind of just big stars with the axons and dendritic tree all being reasonably similar. So morphologically, you can't really distinguish them. You can't really distinguish the directional flow of information through that neuron. Next is a bipolar neuron. A bipolar neuron has one dendritic projection that is for the incoming information and the input. Then the graded potential passes through the cell body, and the axon emerges from the opposite pole of this neuron. So you can imagine, just by comparing the structure of an anaxonic neuron and a bipolar neuron, that the specific characteristics of the graded potentials that are going to be generated on the different dendritic projections here is going to be qualitatively different, right? So whatever graded potential has to come into the dendrites of a bipolar neuron is going to have to be significantly stronger than would be present in an anaxonic neuron. You can have summations about an anaxonic neuron, the various dendrites in the anaxonic neuron that don't have to travel down a long dendritic projection before they can elicit an action potential at whatever the axonal output of that neuron is. But bipolar neurons, interestingly, that whatever electric field or graded potential is being generated in the bipolar neuron, that electric field has to pass, or the local currents is another way to describe it, those local currents have to pass through the soma of the bipolar neuron, whereas in a unipolar neuron, they look very similar in structure in that there is a single dendritic projection and a single axonal projection. However, whatever graded potential is elicited in the dendritic projection, that accompanying electric field and local current of ionic flow doesn't actually pass through the cell body. And you may say, well, what's the importance of that? Well, that does require a deeper discussion of sort of the biomolecular structure of a neuron. But suffice it to say that the local currents and the accompanying electric field that is generated by them have certain effects on macromolecular, specifically cytoskeletal elements within the neuron. And those effects will be present in a bipolar neuron, but minimized in a unipolar neuron, right? So that the local currents don't really pass through the cell body. A unipolar neuron is indeed sort of just a linker. It links one synapse to another distal neuron in another part of the brain. And then finally, the very sort of the epitome, the archetypical neuron that you see drawn everywhere is the multipolar neuron. This is far and away the most common neuron that you'll find in the brain. And this just has a bunch of dendritic arborizations that are directly attached to the soma, and then a long axonal projection that extends off that soma at the axon hillock. All right, so the point here is that there are these various structural variations on the theme of dendrite, cell body, axon that have functional consequences. There are functional consequences of all of this, and a deeper discussion of which is well beyond what we have time for here. But on to a class of cell called the neuroglia, or glial cells, G-L-I-A-L, glial cells, or neuroglia. So glial cells are essentially support cells of one sort or another. They are non-excitable tissue. They are not neurons. They are not sending signals. They are not receiving signals. They are, in some way or another, supporting the function of neurons. So the ependymal cells are a type of cell that lines the ventricles of the brain and the central canal of the spinal cord. We'll find out what those mean, but essentially the ventricles Gesundheit. The ventricles and the central canal are what you could consider to be the holes in your head. There are void spaces in your brain, believe it or not, and those void spaces are lined with these ependymal cells. They support what are called the choroid plexus. We'll talk about that in a bit, but this is the part of, these are the cells that produce what's called cerebrospinal fluid. Now, your brain is floating in its own kind of aqueous primordial broth called the cerebrospinal fluid. We will talk about it in a bit, but it's produced, circulated, and monitored by these ependymal cells. Next are the astrocytes. Now, I just said that the brain is floating in this broth of cerebrospinal fluid. The brain obviously needs needs blood, or more specifically, the contents of the blood, nutrients, oxygen, etc. But in fact, there's a lot of stuff that's in the blood that the brain does not want to come in contact with. The brain is a privileged tissue that is in some ways held aloof from the brain. There is a cellular barrier that keeps whole blood out of contact with actual brain tissue, and this barrier is called the blood-brain barrier. The astrocytes are one of the mechanisms by which the brain is able to isolate itself from the blood. They also provide structural support. Now, when I say astrocyte, I perhaps unwisely used the word star when I described an anaxonic neuron, the last slide. That is a neuron called an anaxonic neuron. And I said it looked like a star. Astrocytes also look like stars, which is why we call them astrocytes, but they're not neurons. They are not neurons. They are support cells, all right, that look in some ways similar morphologically to anaxonic neurons, although they're much more common than an anaxonic neuron would be. They have all these projections that come out from them in this three-dimensional pattern that connect to various neurons and to capillaries and give the brain its three-dimensional structure. They create the three-dimensional lattice that the neurons find themselves in. The brain itself is obviously highly complex, highly interconnected three-dimensional network of synaptic interconnectivity. And to maintain just the spatial architecture, like the genuine tissue level architecture of all those connections, you have to have some kind of scaffolding to support those neurons. That's provided by the astrocyte, amongst others. Oligodendrocytes also help provide this three-dimensional architecture. And the oligodendrocytes, the primary job of them is to provide myelin. So remember when we talked during the cell physiology lecture about myelin, which is essentially the insulation that you find around axons that helps speed up synaptic transmission? Well, in the central nervous system, this myelination is achieved via oligodendrocytes. What's interesting about oligodendrocytes is that in contrast to the peripheral nervous system and the Schwann cells, which do the myelination there, in the central nervous system, oligodendrocytes, a single oligodendrocyte or single cell is going to have multiple, or oligo is the prefix that refers to several, oligodendrocytes. So they are cells that have multiple dendritic projections that come off of them. A single cell is going to have several projections off of them that are going to myelinate a number of different axons on proximal neurons that may be near one another in some sort of tract in the brain. Now, can you guys, I've already kind of given you the answer to this, but just to see if you can put it together. Why would that be? Why wouldn't an oligodendrocyte just myelinate a single axon, a single cell? Why would it be potentially advantageous for it to myelinate a number of nearby or proximal axons on various neurons? What do you think? I've already said it really today, but what's one of the characteristics of the brain tissue that I said earlier? So I'm showing you pictures of the brain projected on this two-dimensional screen, right? But is that actually how the brain works? What do you think? Take a stab at it. Why is it advantageous to have an oligodendrocyte have projections that attaches to multiple axons? Take a chance, somebody. I'll just sit here. Nobody? Come on, somebody. Absolutely, yeah. Adding as much myelin as you can, making sure that these cells are completely myelinated is important for the speed of the transmission. But I'm specifically asking, why is a single cell having multiple projections between adjacent neurons? What did I talk about in terms of the function of the astrocyte? Yes, it's just another part of that scaffolding, all right? It helps neurons maintain their three-dimensional structure with respect to one another. Okay, does that make sense? All right, and then lastly, microglia. Simply the dump trucks, the garbage trucks of the brain. They're going to take care of any waste materials or debris, cellular debris. Say there's a cell dies for some reason. And if by any chance there is a pathogen, it's going to be picked up by these phagocytic cells. What's a phagocytic cell? You guys remember? I talked about macrophages at one point. Yeah, what is it? Phagocytosis. Good. Yeah, so that's a complicated word. That means eating. They will just surround and absorb and break down other cells, so pathogens. If I'm using, you guys got to let me know if and when I'm using a word and assuming that you understand that word, because I've been teaching this stuff so long that I forget, like the word is really familiar to me because I've said it so many times that I forget that you haven't heard that word, maybe ever. And if you haven't ever heard that word, you just stop me. Don't ever be shy. Please, this is a great opportunity in here for me to not be judgmental of you so that when you go somewhere else out in the world and some other person is blowing a bunch of words past you, you're not afraid to say, hold on, what does that word mean? Tell me what it means. Yeah. Don't ever be shy. Please, please. Ask a question. Your peers obviously have the same question. All right, so neuroglia also exist in the peripheral nervous system. There's not as many categories. There are these satellite cells, and the satellite cells are mostly found in what are called ganglia. Ganglia are clusters of cell bodies that exist. There are clusters of cell bodies that exist in the peripheral nervous system someplace. Usually around some part of the autonomic nervous system. They're going to help to regulate the chemical environment in those ganglia. So oxygen, nutrients, waste, they help turn over neurotransmitters that may be accumulating in the ganglia. So what's a ganglia? Cluster of cell bodies. Cluster of cell bodies in the peripheral nervous system. A little ball of nerve tissue, mini brain, so to speak. And then Schwann cells are the myelination of the peripheral nervous system. Now, why are Schwann cells, why does a single Schwann cell form a single internode, a single myelinating component? Why doesn't it attach to multiple neurons that are adjacent? Can you guess? It doesn't really need to form a three-dimensional network because in the peripheral nervous system, most of the axons that are adjacent to one another are just tied together in a bundle by the connective tissue of the central nervous system. We probably won't get a chance, looking at the time, we probably won't get a chance to talk about that connective tissue sheath that's in nerves, whole nerves in the peripheral nervous system. But it doesn't really need to have this complex three-dimensional architecture where axons could be traveling at weird angles to one another in some complex part of the brain. Right? So a single Schwann cell for each internode is sufficient. All right. Here are the 6 regions of the brain. So there's a little bit of anatomy. We don't actually need to go through this anatomy in gross detail here because what's important? I'm just going to need to talk about a couple of the things. So the cerebrum, the big cauliflower part of the brain that is the region that most people think of when they think of the brain, that is all of the higher order functions of the brain. So for example, complex reasoning, strategy, memory, and not base consciousness, but conscious thought process like language, all this kind of stuff. And importantly for the discussion today, conscious control of skeletal muscular function and also the somatotopic mapping of your sensorium. So sensorium means the sensory experience. So there's this internal representation of the external world that is built out of the information that all your sensory receptors gather, and it's mapped onto parts of the brain, right? And I talked about somatotopic mapping when I talked about taste, for example, and the labeled line code and how that goes up to the brain. But we'll see how that plays out, hopefully, at the end of the hour tonight. All right, what else is important here? We will talk about the thalamus quite a bit. So the thalamus and the hypothalamus and the diencephalon, they're part of what are called the basal nuclei in the brain. The thalamus, for you to be consciously aware of anything, any kind of sensation, there has to be a synapse in the thalamus. So any sensory input that's telling you any kind of thing, hearing, sight, whatever, feeling, touch, all that, it's going to have to synapse in the thalamus before it projects up to the cortex. And then the hypothalamus will be more important when we get into the endocrine chapter. It is kind of the bridge between, so I said the pituitary gland or the hypophysis was sort of the king or queen, the top of the pyramid in the endocrine system. But the hypothalamus, which connects to the pituitary, which is that little, this little berry that's hanging off the bottom here. The hypothalamus is this region of the brain that connects the rest of the neural tissue, the brain, to the endocrine system. And the pons and the medulla are important for the cranial nerves, and I think that's all that you actually need to know in terms of discussions we'll have. Here are those holes that I talked about in the brain. These are important because they are filled with cerebrospinal fluid and are responsible for the circulation of the cerebrospinal fluid. So there are these void spaces. The blue that you see are void spaces in the brain that then communicate with this thing called the central canal that runs down the middle of the spinal cord. Inside of it is cerebrospinal fluid, which is this broth that I talked about that's produced by the choroid plexus, these specialized ependymal cells. And it is resorbed by what are called the arachnoid granula. I'll discuss those in a moment when I talk about the meninges of the brain. The meninges are the connective tissue support structure of the brain. So what is it? What does the cerebrospinal fluid do? It adds buoyancy to the brain. It helps the brain not just sort of like sit like a big bowl of gray jello in the cavity of your brain. It adds some buoyancy to the brain, helps to lift it up into the cabinet of the brain case. And because of that, it also protects it. So say you have, God forbid, any of you, athletes or otherwise out there, have fallen down or gotten hit on the head, scary injury to me, that brain and the cerebrospinal fluid keeps the brain from slamming against the wall of the brain case, which is, your muscles can bruise and recover, but the brain is a much more delicate organ than that. And then, of course, it gives it the chemical stability that's required for a complex organ like the brain. So you're going to have all the nutrients, the chemical messengers and waste products. That's all a function of the cerebrospinal fluid. All right, on to the meninges. So I should probably put. There's a lot of text on here that is probably not necessary. There are 3 layers of meninges. This is what's important. 3 layers of meninges. And going from superficial to deep, they are the dura mater. You can see that up there on the left. D-U-R-A, dura mater. The dura mater is so-called because it's the durable mother. Dura mater is Latin for durable mother. It is the tough, thick connective tissue sheath that is the durable sheath that forms this connective tissue sheath around the entire brain for its own protection. All right, the durable mother, the dura mater. The next one down is the arachnoid mater. arachnoid mater. You can see that label below the dura mater there. Arachnoid refers to arachnid. What is an arachnid? Spider. So the arachnoid mater is the spidery mother, kind of creepy, but it's just a description of what it looks like. The arachnoid mater has these spidery tissue tendrils that underlie the dura mater and connect it to the brain, sort of giving a loose, fibrous support structure. The arachnoid mater is also where we have what are called the arachnoid granulations. So I said that the choroid plexus produces cerebrospinal fluid, and the arachnoid granulations resorb it. If the arachnoid granula did not work, you would keep producing cerebrospinal fluid, but you would not be resorbing it, and the intracranial pressure would go up. What do we call that? Do you guys know what that condition is called? Meningitis is an inflammation of the meninges. It is a problem with the meninges, but not this problem. What's it called when you can't resorb enough cerebrospinal fluid in the brain and there's a lot of fluid that builds up in the brain? Yeah. It's hydrocephaly. Yeah, it's hydrocephaly. It's a dysfunction in the arachnoid granula, and you get an increase in blood pressure, the ventricles swell, it compresses all the neural tissue. Those people have a lot of problems. You just put a little shunt in the brain, drain the fluid off. These people lead reasonably normal lives. Then lastly is the PIA, P-I-A, PIA mater, the sweetest of them all. PIA just means little mother, the little mother of the brain. And it is the PIA mater's duty to be just a thin tissue membrane that lays directly on top of the surface of the brain and kind of pins the capillary structure of the brain down onto the surface of that, the PIA mater. All right. Here we can see the flow pattern. So in the center of your brain there, you see that red tissue in the ventricles there, the third and fourth ventricle. And it's producing this choroid plexus is producing cerebrospinal fluid that circulates around the brain down through the central canal, out around the outside of the brain, and is eventually resorbed by these arachnoid granula that are connected to the venous structure of the brain, what's called the superior sagittal sinus. But that's anatomy that you're not necessarily responsible for, but there it is. All right. How is that? Everybody cool so far? Blood-brain barrier. Yeah, I could probably have skipped this slide. I guess I already had talked about this a little bit. So the blood-brain barrier is, as I said, this barrier that gives the brain a sort of exclusivity to the contents of the blood. It's formed, first of all, by capillary epithelium, just the epithelial cells of the capillaries. It keeps the blood off the neural tissue. But that capillary epithelium is in contact with special ependymal cells that are specialized for this purpose. And then astrocytes. So astrocytes, the projections off these astrocytes surround the capillary epithelium and allow the passage of only a very select range of solutes, such as the dissolved gases, such as nutrients such as glucose or ketone bodies, whatever it happens to be. When you doctors do a diagnostic process that I'm sure you've heard of called a spinal tap, not a parody heavy metal band. It is a procedure whereby they remove some of your cerebrospinal fluid and they look in it for a number of different solutes. But if your protein level is elevated in the cerebrospinal fluid, or worse, if there's cellular components in there, like red blood cells or white blood cells, this indicates that there's been some sort of breakdown in the blood-brain barrier. And there may be due to inflammation, maybe meningitis, maybe, who knows, a tumor or something. But the blood-brain barrier, it isolates the neurons. Here are the 12 cranial nerves. And sadly, this isn't really an anatomy class. I'm not going to get a chance to go through them in detail. They're pretty interesting. I enjoy talking about them. But I'm going to point out just a few of them because on Monday of next week, we're going to start talking about eyes. But so that you can understand the cranial nerves, I said that a cranial nerve was a nerve that comes directly off the brain. So it communicates with the periphery by directly emerging from the brain, not the spinal cord. There are 12 of them, and they're numbered by how they emerge from the brain case, anterior to posterior. This is anterior right here. We're looking at the inferior surface of the brain. Here is the olfactory bulbs, for example. This is your sense of smell. The next one is cranial nerve 2. This is the optic nerve. This is the nerve that sight is transmitted along. Then there's a host of ocular eye muscle nerves and the trigeminal nerve, which is not important for our discussions at all. The next one that actually is important is cranial nerve 8. Here it is, cranial nerve 8. This is the vestibulocochlear nerve. There's two branches, the vestibular and the cochlear branch. We'll talk about those on Tuesday when I talk about hearing. So hearing is transmitted by one of the cranial nerves. And then another one that's going to be important later is cranial nerve 10, the vagus nerve. And the vagus nerve comes off of the pons. The pons is responsible for, it comes off the pons. The pons is responsible, it comes off the medulla oblongata, pardon me, I was wrong. The centers that originate, give rise to the vagus nerve, regulate heart rate and respiration and the digestive system. So in each of those different chapters, we'll talk about the vagus a little bit. None of the rest of them are going to play into any discussions that we have throughout the semester, I don't think. The glossopharyngeal and the facial nerve, 7 and 9, I'll talk about a little bit when I talk about taste, because they're responsible for taste and salivation. Anyways, there's the cranial nerves. Descending the central nervous system, we get down to the spinal cord. So we're going to need a little bit of spinal cord anatomy to be able to have our discussion of sensory and motor pathways. So when you look at the spinal cord, there's gray matter and there's white matter. The brain in general is made of gray matter and white matter. Does anybody know the distinction between gray matter and white matter? Yeah. Actually, just the opposite. Yeah, but you're right. You were right. You just had it backwards. So white matter is white because it has all this myelin wrapped around it. Myelin is just the cell membrane of the Schwann or oligodendrocytes. In this case, since it's the central nervous system, there'll be oligos. But membrane, what's membrane made up of? Lipids, right? Phospholipids. And lipids are another name for fat. So really, white tissue is just, it's white because there's a lot of fat molecules, lipids in it. And then gray matter is just the cell bodies. All right, so there's usually synapses. Synapses happen in gray matter, and white matter are just axons that are myelinated that are projecting one way or another. Okay, so in the spinal cord, there's going to be synapses in the gray matter, and then there's axons that are going to travel up or down along columns or tracts in the white matter. The spinal cord is broken up into all these nuclei, which are clusters, functional clusters of cells. And those functional clusters of cells, they're highly organized. And in general, anterior, which means if you're thinking about your spinal cord deep, right? Anterior is deep. Anterior is up here. This would be outside. This would be in towards your body. Anterior is going to have, these are motor nuclei, so efferents. And then posterior, these are all sensory nuclei. And that's afferent. All right. So the spinal cord is broken up into all these various segments that correspond with the vertebra, and the nerves emerge between each vertebra at these different segments. What do I want to say? Often these spinal nerves form what are called plexuses. A plexus is just a complicated, interconnected network of nerves, a complicated meshwork of nerves. There's several plexuses. I used to go through them all. I'm only going to show you the cervical plexus. And the only point I'm going to make about cervical plexus beyond giving you an example of what a plexus looks like is that the cervical plexus gives rise to a really important nerve called the phrenic nerve. It comes off of C3, C4, and C5 nerve roots. The phrenic nerve goes down to the diaphragm. The phrenic nerve goes to the diaphragm, the thoracic diaphragm. So if the phrenic nerve gets broken or the spinal cord gets cut by an injury of some sort above C3, then it's life-threatening because you are now no longer able to breathe. So neck injuries at C2 or C1, those, if that person's not stabilized very rapidly, they will be unable to breathe and die. Right. So below C2 or C3, below C3, C4, C5, they can survive. They will just become paraplegic or quadriplegic. Paraplegic if it's above what's called the brachial plexus. Quadriplegic if it's above the brachial plexus, so they can't use their arms, or paraplegic, just their legs, if it's below the brachial plexus. Yeah, it's important to know the nerve anatomy of the neck. All right, on to the spinal tracts. I think I might make it today. We'll see. So there are these, I showed the nuclei of the gray matter. The white matter is these, there are columns. There's a posterior, a lateral, and an anterior white column. And these columns themselves are broken up into tracts. You don't need to know the names of them specifically at all. That's for a neuroanatomy class. But you do need to know that ascending tracts, and I'm showing them ascending on one side and descending on the other. That's not how it actually works. There's ascending and descending tracts on both sides. I'm just separating them out for clarity here. But ascending tracts are going to be afferent, right? They're going up, and that's going to be carrying sensory information. Descending tracts are ones that are carrying efferent information or motor information. They're descending from higher structures outward. A couple bits of vocab. Decussation is when a fiber goes from one side to the other. It crosses the midline when a neural pathway crosses the midline. It's called decussation. And then the directional terminology. Contralateral is a relative term, meaning on either side, opposite sides of the midline. So my left ear is contralateral to my right nostril. Oh no, it's like this. and ipsilateral means on the same side. So my left ear is on the same side as my left eye. They are ipsilateral to one another. Right? But those terms are important with the brain, because the brain is divided into two halves, and the nervous system in general is divided into two halves. So we often have decussation, and often you can have symptoms that are contralateral or ipsilateral, and that tells you where the pathology or lesion might be somewhere in the nervous system. Okay. Sensory pathways. I'm getting close to where I wanted to be. So these pathways, these ascending pathways, involve three neurons. Sensory pathways involve three neurons. The first order neuron is the sensory neuron. It's the sensory neuron. It's some sensory receptor, and it gets triggered. The signal comes back, the cell body of which lives in what's called the dorsal root ganglion. So when I was showing those pictures of the spinal cord, I didn't really show this ganglion, I didn't point it out, but on the dorsal side, on the posterior side, there's this anterior and dorsal root. The dorsal root has a ganglion associated with it. That's where the cell body of the first order neuron is. Comes in to one of the sensory nuclei in the gray matter of the spinal cord, and it synapses on a second order neuron. The job of the second order neuron is to just go upstairs. It just ascends. It ascends to the thalamus, where it's going to synapse on the third order neuron. It's going to synapse on the third order neuron only if that sensation is one in which it's going to register in the body, in the brain somehow, that you're going to be aware of it. Right? If it doesn't synapse in the thalamus, you don't know it happened. All right? So tumors or strokes or some kind of lesion in the thalamus can be pretty disorienting and disruptive to that person. Reflexes that happen, so say there's a muscle stretch reflex, that sensory neuron comes in, synapses on the second order neuron, and it can have a collateral that comes out and synapses on a collateral branch that synapses on a motor neuron without having to ascend to get a stimulus to elicit the reflex response. All right. And then third order neuron is simply the neuron that projects up to the cortex, to the somatotopic, somewhere in your cerebrum. All right. Sensory pathways and Wilder-Penfield. So this guy is one of my favorites. He's such a, in science, science for so long, even still, maybe this is a reflection of the world at large, can be plagued with egomaniacs and jerks. Like, it is just full of people that are not super pleasant sometimes, right? And that are not especially collegial or anything. This guy is a beautiful contrast to that. He was a really lovely person by all accounts. I never got to meet him. He was dead before I hit the scene. But I did read his book and found it very engaging. This guy, he's from Canada. He went to McGill. And then, no, I'm sorry. He was a researcher at McGill back in his home country, but he studied at Oxford with William Osler, and then for undergrad. No, he went to Princeton and then studied at Oxford, and then in New York with Harvey Cushing. Osler and Cushing, for those of you who go into medicine and are interested in history at all, those are some of the great physicians of the 20th century. So he was highly pedigreed. While at McGill, he was a brain surgeon, and he would do the interesting thing about the brain is that when you operate on the brain does not actually have pain receptors in it. There are not sensory pain receptors in the brain. And so you could have brain surgery on a patient while they were awake with local anesthetics around the point of calvarectomy. So they could like cut a big chunk of cranium off, fenestrate the brain case, reveal the brain, and take out the tumor or whatever. And while he was in there, you could talk to the patient while you were operating on their brain. And he would be cutting out a tumor, and the patient would say, "I smell almonds. I smell almonds," or "My nose itches now," or like, the hand would start doing something or whatever. And so this was the guy that first did the kind of, at this point, cliched or cartoonish thing where you're like sticking electrodes in the brain at different places and eliciting all these responses from the patient. He was able to do that. He mapped out these somatotopic maps in the brain that relate to various sensory aspects of the brain and motor aspects of the brain. He was the first person to do that. And he developed what's called the homunculus, which is something that's important that I will have to talk about on Monday because I can't let it go. I'm still chasing that 15 minutes all semester long. On Friday, I will see you guys here. Please figure out what time you're coming to meet me. You can find the sign up there in Moodle.